Such a mood. Hopefully Zoom doesn't screw up this time and like split us into 2 groups. So yeah, let's hope not. But if any of you guys have like friends who can't get in, just let us know as soon as possible, because
Zoom sucks.
Hopefully we'll avoid what happened last time. Yeah, not keen on that. It's so stressful. Yeah. So I guess how tonight will work. You guys can either use the chat or you guys can just unmute yourself, I think. I think it should be okay. Just use common decency. But how do you guys find week 2.5 or like week 2A, 2B? Like, was it a mess or was everything pretty okay?
I heard the toots were bad. Like, they were all over the place.
Oh, yeah.
Yeah.
Yeah, I kept seeing the posts. That Zoom, but it's not working. Were there any topics that anyone wants to include in particular or for us to spend more time on? HKS, Clin Skills, Biochem, genetics, ethics, things like that? Or do you want to just go with whatever?
Whatever is highest yield.
That's fair.
Oh yes. What topics are you guys interested in for more quizzes?
Oh yeah, true. Whatever's highest yield. Okay, that's fair. Well, we always do high yield things.
Yeah, we only include high yield in our PowerPoints.
I think someone said cell cycle regulation for the quiz.
Cell cycle regulation. Oh, like
Okay.
more of the cyclin CDKs.
Oh yeah,
but
I added in some last night.
Oh,
that's
that's
not
good.
very high yield.
Yeah, that's high. It's like all right.
You get like one question. Oh, sorry guys, 1 second.
What was the active learning?
Active learning was on Central Dogma, but like the extra stuff.
Was that the French guy? He like divided you into 4 groups.
It was the extra booklet, and then they told you about like
But we had this on Zoom, so it's like super like uncoordinated stuff.
Oh yeah.
Like imagine being in groups and then like reshuffling, but doing that on Zoom.
It's
Bro,
like
confusing enough already.
Confusing in real life. It like didn't make any sense in real life, so I assume like Zoom just made it even worse.
Yeah. So Enoch, that was like socialization and like ubiquinone and things like that.
Today, unofficial revision group.
Yeah.
It's anyone can join.
Yeah. Okay, what else? Active learning. Do you guys have another active learning this week?
Not too sure. Oh, yeah.
Doesn't sound that useful then.
We have like the great metabolic race.
Oh, Norma!
Oh, great metabolic race.
I love Norma.
Actually, last year they said the great metabolic race active learning was like not compulsory, so like no one showed up. But then it turned out to be like one of the highest yield sections for metabolism. So like do learn integrative metabolism. That's where all the questions come from.
That's true. L, yeah. Yeah, I remember that's like when a ton of us went out for hot pot right before the Great Metabolic Race because
yeah, that was cancelled.
So oh yeah, and I didn't go. I just didn't go.
Yeah, I didn't go to active learning.
We went and got bubble tea instead.
Yeah, just went home, I think.
Guys are really studious. Yeah, we really like value you studying.
High yield. Because they said it was not compulsory, so I took not compulsory to mean like non-examinable.
They said it was optional. Like literally. Like Nirma, I like heard those words come out of her mouth. She was like, it's optional.
Can we start?
Yeah, do you want to start?
David, stop plugging your Instagram.
Don't worry, we'll be talking about David's Instagram in the Kahoot quiz.
Are we?
Yeah. I was up so late last night, just like adding stupid questions into the week 1 quiz. That's why I texted David at like 2:30. Just like, David, how many texts?
So late last
David,
week.
how many IG accounts do you have?
You shall find out soon.
Yeah. So we'll be getting started. So since the earlier session earlier this week got absolutely destroyed, we'll be going through week 3 again. So
do you want to do the week 1, week 2 Kahoot first?
Oh yeah.
Oh, that's a good idea.
Good engagement.
Yeah, let's do that.
Oh my god, guys, 70 people. Hi everyone.
Who wants to share screen? RJ, do you want me to share screen or?
Yeah, I can share screen.
Okay, RJ will be sharing screen. Get your Kahoots up. Every 5 questions, there'll be a joke question, I think. If I can count.
Wow, Peter, you put a lot of thought into this.
Right, I did. Right, like, I was so cooked at the end of it. I was just like, oh, what's another joke question I can put in? Yeah, so hopefully it's not too bad. And what we've also done with the Kahoot quizzes is that we've moved them all onto a Word doc as well. So you'll have the questions as a Kahoot quiz form, as well as a Word doc, so you can go refer back to it when you're doing revision, or if you want to check out the answers afterwards. And obviously they have the answers attached as well.
Okay. How do you share the sound?
Oh,
No, it's fine, right?
Oh yeah, how do we?
I'm pretty
if you
sure.
unplug your earphones and you turn your volume to max, it might work.
Oh yeah, let me just try again.
I think it's in one of the settings, RJ. Like when you select the window or select the screen, you can choose to share the computer audio as well.
Also, guys, PG-13 names, please. We keep it, you know.
This is recorded.
Why would you say that, Heini? Now it's gonna go crazy.
Yeah, now it's gonna be like Happy Cat 3.
Also, guys, feel free to like turn your video on. Like, we want to see all of your faces.
Can you hear my?
Just slightly strange.
Oh yeah, so we're bumping.
Can you hear the sound now?
Yeah.
All right.
Maybe turn it down a bit.
Enoch's boyfriend. Oh yeah, Enoch got a Monash love letter.
Dude,
Congratulations. Pingu's boyfriend. Oh, what the heck?
Wait. How do we tune this down?
Can you turn it down a bit?
RJ, turn it down.
Hello. I already lowered it. Yeah.
Okay, good.
Nah, that's better.
That's good. It's background music.
Yeah, it's bopping.
By the way, how many emails do you have?
What do you mean, how many emails?
Look at how many emails you have.
Jating, don't try to
I'm
get
doing
how many
me?
IG accounts
Wow.
David has.
Oh, right.
Yeah, I see your tactic. Alright, just waiting for a couple more people, I think. And then we'll get started soon.
So how many people are there? 70, right?
There are 60, high 60s in the Kahoot now.
Isn't in the
Zoom call, there's
Zoom call.
76, and that's chaos.
So, like, excluding... How many invites are there? I can't count.
There's 5.
5? Okay. So we should have 71. Right? No.
Come on, family guys.
72 now. Oh, what? No, 73.
Might be. Hey.
I love gnocchi. Oh, bro, that's such a mood. Who's I love gnocchi?
Yum. Who's Pingu's
Wait, I'll
dad?
join. Give me a sec.
Wait, yeah, I'll
Guys.
join as well. Wait, should I join? No.
Guys, I don't want to join because it'll reveal that I don't actually know any of the year 1 content. So
Yeah, bro.
it's gonna be a bit of a shock.
I love Gnocchi.
Gnocchi.
Gnocchi. I am lonely. We love you. I am lonely.
Okay, so shall I start?
Wait, we have 80 people
Oh,
now.
Oh, we have 80 people. Come on, guys. Join the Kahoot. We'll be
Come on,
getting
let's
started.
join and then we'll get started. Cheerful quota.
Is that it? Q. Rapid shock 45. Alright.
Let's go.
start?
Yeah, let's just
Okay.
start. Let's start.
Yeah, I'm gonna start. 3, 2, 1.
Go.
I am ready.
Oh, no,
Oh,
the
bro,
Pingu
these
got
questions.
cut off.
Oh, that
proud.
was the old PS Pingu logo.
Yeah, bro.
Oh, doesn't matter.
Not lovely. Come on, guys, speed. We're all talking about that
And
speed.
speed. Remember how fast our week 1-2 PSP
I am
session was?
speed.
I expect the answers to be as quick as that.
Hey, RJ, you might want to turn the volume down a bit more if you can.
Oh,
What's the lowest?
very
Oh,
nice.
okay.
Should we go
Oh,
through
okay.
these questions really
Let's go
quickly?
through, yeah.
All right. So, original cell theory. How do we want to go through it?
It's not really important for you to know like the difference, I guess, between because modern cell theory is basically original cell theory, but it's just like scientists like figured out more stuff and they were like, oh, okay, we're just going to call it like modern cell theory, but it's just like the same with just like more elaborated points. But yeah, so the most basic kind of point to take from the original cell theory is that a cell is the basic unit of life. And so, yeah.
And also you need pre-existing cells to give
Yeah.
rise to new cells.
Okay. Next question.
If you have questions, type them in the chat as we go.
Good job, Kirthana. These questions are also mixed up, so it could be like week 1 and then week 2 and then jumping back to week 1.
Yes.
Yeah. Okay, very good.
I feel like that one's like pretty self-explanatory. Like, do people like get that one?
Is everyone okay with it? I'll just move on since the majority got it right.
Oh, also a point with that is that Med loves to do that. Is like they say which one is false or which one is like not correct, and like the first thing you pick is like you always think it's like the whole like reverse psychology thing. Like Med always does that in their exams.
Yeah, I think they do that still for Sem 1, but for Sem 2, they've cancelled that because Ian Presnell, like, you guys will, like, find him later on.
Yeah.
Yes,
Yeah.
he was, like, very against those questions, so I think he removed it. But for now, like, you might still have a few of those.
Yeah, I had like I like half broke down because I was like trying to write a question last night and it was like using negatives and I was like, what's going on? I feel like I would get this question wrong again. Yeah. Cancel reverse.
How did people go?
I think we should go through this.
Yeah, let's go through this one.
Okay. Let me see if I can. Oh, can I share at the same time?
Should be able to, I think. Yeah. You can share.
Okay. Okay, can everyone see that?
Probably
Thanks,
not.
Christina. Can people see that? Can see my screen?
Yeah.
No, we still see the Kahoot, right?
If not,
So you
go
need
up
to
to the
change the
top.
view options.
So you go view
Oh,
options and you click on Heine instead of RJ.
Yeah.
Is at
Can
the
you just
top.
stop my share and then you can share yours?
Wait, I need it. What was the question though?
It was,
It's like all of it.
yeah, just go through
Okay.
microtubules, microfilaments.
Microtubules. Okay. Let's just go through this again. So, basically, as we know, there are three main things that you guys need to know about the cytoskeleton. So you've got microtubules, microfilaments, and intermediate filaments. Oh, sorry. Wait, let me keep the chat open. Yep.
Their functions.
I'll just go into it. So basically, microtubules, they maintain cell shape and they control transportation of cell substances in and out of the cell. And they also help the cell resist any compression forces. So basically just like keeping it in that kind of structure, even though like, you know, we did talk about like how cells are kind of like fluid in a way, but like you also need it to have that structure so it doesn't like go everywhere. We did go through how it is involved in mitosis. So basically, the microtubules are part of the mitotic spindle, which help pull chromosomes apart. But that's more to do with cell cycle. So, also, the other thing is cilia and flagella. That was in the previous question. But cilia, can I add a diagram of what they look like? No, I didn't. Okay, that's it. Okay, basically cilia are like so like if you've got like a flat surface, it's like little tiny like protrusions like along the surface, but like flagella is like a big long tail. So like that's why with the previous question, how sperm was the wrong answer, that would actually be flagella, not cilia. And so basically cilia, it's especially common in like, especially like the respiratory tract. So it helps like, it kind of just like moves and like waves and stuff and like helps the mucus like move down. And both of them have the 9+2 arrangement, which you can see here, which is very important. You do need to remember the 9+2 arrangement. Centrioles, that's, yeah, so they're involved in the spindle formation during mitosis, but that's more like cell cycle kind of stuff. Okay. Do you want me to go through all of it or just the microtubules part? Anyone have anything there? All of it? Okay. So microfilaments they include two different types. So you've got actin and you've got myosin. So actin is the one where you've got like a lot of it. So they're arranged in an intertwined double helix, and there's a high concentration of it at the cell's periphery. And myosin is basically, you don't see much of it at all unless it's in muscle cells. So you can't actually see them under a microscope in normal cells. And then we'll go through arrangements. This is basically just taken directly from the lecture. Hello, someone said something. Yeah, so, okay, wait, no, I'll finish this and I'll go back to talking about that. So some arrangements, so you've either got them in skeletal muscle, and then you've got them as the actin sheath beneath plasma membrane. So that helps with the transport of substances and cell movement. And then 3, we've got movement of cytoplasmic organelles, vesicles, and granules. 4, we've got cleavage in mitosis. Basically, it helps like pinch. So when you've got like how it's like becoming 2 cells, and it helps pinch the cell into 2. And then you've got random and unorganized. So basically it's just like scattered in the cytoplasm. So a lot of it's actually like to do with like, basically main things are cell structure, cell movement, and just like transport of substance. And I will come back to your questions. Don't worry, I will come back to them. Intermediate filaments. Basically, this is the one where, like, you can see in the table here how there's like lots and lots of different examples of intermediate filaments. Don't go trying to like memorize a lot of the different, because like there's huge tables that of like the different examples. I would just say no, maybe 2, just so you know like kind of what they're involved in. But basically, once again, it's helping to maintain cell shape. It helps like anchor nucleus and organelles in the actual cell. And they're made of multiple strands of fibrous proteins. But yeah, please don't go and memorize like a lot of different kinds of like intermediate filaments and all that kind of stuff. It's just, it's not worth like your brain. Okay, wait, sorry, I have to go like back. Oh, thanks, Peter. Yeah, 9 plus 2 arrangement. I'll just go back to this. That's just this over here. So I think people answered that one though, so that's all good.
Yeah. Cilia and Flagella have the 9-2 arrangement. So 9 on the outside, 9 dimers on the outside, and 2 on the inside.
Okay, is that everything? Because I think everyone else kind of.
I'll link the Google Doc.
Oh, do you mean the slides?
The PowerPoint? Yeah, I'll do that, Heidi.
Okay, thanks.
So carry on with the code.
Yep. Does anyone else have any questions?
You don't need to know exactly how cilia does cellular signaling. That is just a small function that you don't need to focus on at this level. Answering a previous question.
Okay. Cute.
All right, RJ, take it away.
Okay. Yeah. Okay, next question. Let's go.
Who's on top? Jen's on top, and smiley face. Knocky.
Yay!
Okay, next one.
Good job, Jen. Alright, there's a joke question after this. I know what this is. Alright, very good. Good job, everyone.
All right. Okay.
Yeah.
Next question.
Oh, Pinder's dead. Jokes. That's actually not a real code. All right, RJ, after this question, we'll go back and explain the past few questions.
Yeah, sure.
job, guys. Okay,
Alright,
so we'll go through questions 4, 5, and 6. So question 4 was what happens in late prophase, and the correct answer was the nuclear envelope and nucleoli begin, well, they disappear essentially. So we'll go through why the other options aren't actually correct. So option A was chromatin begins to condense. That's true in some sense, however, it more primarily occurs in early prophase. Spindles growing and attaching to centromeres primarily happens in metaphase, so the spindles kind of help to line them up at the metaphase plate. And then option D, the nuclear envelope reassembling, that occurs in telophase. And therefore that leaves you with the nuclear envelope and nucleoli disappears. And that's for question 4. Crossing over is essentially one of the ways that we get some genetic variation. So during metaphase 1, you have the homologous chromosomes kind of lining up at the metaphase plate. And essentially what happens is that part of the chromosome arms line up over each other, and they essentially swap the alleles or swap the genes from each homologous chromosome. So that allows for some of that. So therefore, it can't be option A. Chromosomes are arranged randomly along the metaphase plate. That describes random assortment. Spindles crossing over and attached to chromosomes, that just occurs in metaphase. That's not actually related to crossing over. And then chromosomes separating and moving to opposite poles of the cell is to do with anaphase. Once again, not to do with crossing over. And then I believe we just went through question 6. Which of the following correctly matches the part of the nucleus and its function? Is that right? Have we gone through that one yet? Yep.
No.
Have we answered that one yet?
Yes.
Yeah, we did.
Yeah. Okay, sweet. So, so a few things is, well, the nucleoplasm as the site of ribosome synthesis isn't quite correct, sorry. Ribosome synthesis actually occurs in the nucleolus, so slightly different. Nuclear pores doesn't serve to keep pH, it more so serves the function to let mRNA out of the nucleus. And then the nucleolus isn't a communication function, it's simply a region within the cell. So that leaves us with the nuclear envelope being a protective membrane around our chromosomal DNA. Does anyone have any questions about those questions? Otherwise, we'll be moving on soon.
Okay. See you next question.
Yeah. Good job, guys. Does someone want to go through this question for us?
Yating, do you want to go through it? Yating, you're our biochem god.
Enzymes essentially only change the amount of energy you need to start the reaction. It doesn't change essentially the energy of the products that you begin, the reactants that you begin with, nor change the energy of the products that you end with. So therefore, delta G doesn't change. It just makes it easier for the reaction to start. But yeah, we're not changing the reactants or the products. So that's why delta G doesn't change.
Mm-hmm.
Sweet. RJ, do you want to take us through the body fluids?
Okay, so basically when you put a red blood cell in a hypotonic solution, that means that water will flow from the red blood cell out into the solution because the red blood cell is hypotonic compared to the solution. And so, water potential goes down a gradient, right? So it'll flow out of the red blood cell and into the solution. And when the cell flows out, it just becomes shriveled. Okay, so whenever you see hypotonic solution, just know that the cell will just lose water. And it won't be ATP because it's all when it's water is always osmosis. All right.
All right, so going through this question here. So this is content that some of you would have gone through in Bio 3.4. But if you didn't get through Bio 3.4, then we'll explain it anyways. So if we consider the blue option first, cholesterol only occurring in cells with a disease state isn't quite correct because cholesterol is actually present in all of our cells, and that's required to maintain the fluidity of our plasma membrane as in the red option. In addition to that, the green option being a key component of transmembrane channels, not quite correct as well, as transmembrane channels are primarily proteins. And then lastly, option yellow, the yellow option isn't correct for the reason being that option red clearly states the function of cholesterol, and that's to kind of stick together our fatty acids when it's really hot, and then to kind of stop them from sticking to each other when it's really cold.
Okay, next question.
Nice, good job everyone. So the reason why it isn't, this one's kind of just a fact. It's simply the structure of DNA. So it's somewhat related to like your organic chemistry from Chem 3.4. So if you number off the carbons on your pentose sugar, the third carbon is on the sugar. However, it isn't indicated by a sugar particle. The 5' end being indicated by phosphate is simply more correct. Yeah. I hope you guys have heard from Richard.
Oh.
We love Richard.
Just random facts.
Yeah. Once you guys meet Richard, you'll probably hear him talking all about Scarlett Johansson. But yeah, Richard is like our favorite guy.
Guys, Richard is so amazing. Like he's just the nicest person.
He jokes about wanting to die very frequently, but
yeah, I know, but it's funny.
Nice. So the reason why it can't be blue, so blue isn't the first step, it's more so the last step, and same reasoning for green. Green is kind of like the middle step, not the first step. Oh, Noki's admirer.
Does anyone want to explain?
If you show the media, RJ, and then I'll also answer Eric's question. Yeah, so the reason why it's not really, why it's not the smooth or the rough endoplasmic reticulum is simply kind of due to the shape of it. You'll start to get used to it over time, but what it tends to be, the Golgi tends to be like multiple pancakes on top of each other that are separated, I believe, whereas the ERs tend to be continuous with each other. Yep.
Layers of lettuce.
Yep, layers of lettuce. That's actually not a bad way to understand.
Yeah, that's
and then Eric, so DNA helicase is involved in DNA replication, so it's slightly different from RNA polymerase. But yes, if we were talking about DNA replication, then it would be the first step, correct? All good. Do you want to take this one? Biochem?
Sorry, what?
Do you want to take this question?
Yeah, sorry, I'm doing other stuff. What enzyme changes pyruvate to lactate? Well, this is more just of a memorization question. You just have to know that lactate dehydrogenase is the one that does the job. I mean, there's not much to say.
Kind of in the name as well, I guess. Like, but
yeah, the only one with lactate in it. So.
All right, nearly halfway there. All right, so we were kind of talking about this before, DNA helicase. So once again, it's talking about DNA replication when during S phase of and yeah, it essentially breaks the bonds between the two strands, breaks the hydrogen bonds, so that the DNA polymerase can get inside. Can't be the red option because one, RNA is single-stranded, so therefore you can't separate it. It can't be blue because that's DNA ligase. Wait, ligase broken strands back together. Oh no, sorry, that just happens naturally to form hydrogen bonds. And then repairing damaged DNA. Repairing damaged DNA is done by a separate group of enzymes. good job. So although the chromosomes start to condense within prophase, they don't reach their most condensed state until metaphase. So it's most useful for them to be in their most condensed state within metaphase because they're about to be pulled apart. So you want everything kind of stuck together before they get pulled apart. Anaphase, although they are condensed at that state, that's not the typical answer for this question. And then during cytokinesis, they're starting to unravel already. So therefore it's metaphase. And that's also where you start to do your karyotyping from chromosomes in the metaphase. Oh, I hope that I changed the answer. Yeah, I think I changed the answer this morning.
I have a feeling I know this. I feel like I know the answer.
Definitely 10.
Surely.
Oh no, I forgot to change the answer. Oh well. David actually has 7 accounts. Whoops. I forgot to change the answer. Oh well. But it's
David, how do you have so many emails?
a stupid amount. David, you're muted.
Yeah, mute.
Some of them are like public accounts. So it's like, for example, AMSA, like the convention that was like going to happen. Like I was running that and like music and stuff like that. So those are all like public accounts.
David's basically a social media influencer. Nice. So G0 phase is that kind of weird phase off the cycle. It's kind of just where cells are chilling out, not doing very much. Cell growth and organelle synthesis occurs in G1 or GAP1, and then DNA replication occurs in S phase. So G0, they're not really doing very much. They're just vibing out.
So all the other 3 are actually modifications after post-transcription. And folding is after you form your primary proteins, and this is after translation. So proteins after that, they fold and form secondary and then tertiary. So it's not actually a post-transcriptional modification. Okay, next one.
Nice. So just a small kind of detail question here. So Okazaki fragments are formed on the lagging strand because your DNA polymerase has to jump back and forth since it only moves in the 3' to 5' direction. And just a small detail with the blue option, it's joined with DNA ligase since we're making DNA and not RNA ligase.
So all the other processes actually occur during apoptosis. Syntactically, is actually something that you'll learn in semester 2. It's just a condition where 2 of your fingers are like fused together. Okay.
Yeah, so originally as you develop, your hand actually starts off as a blob, and the reason why you have fingers is because apoptosis has occurred, and that's how it separated out your fingers. For those who have that condition where they have maybe 2 fingers stuck together, that's because apoptosis did not successfully occur.
Okay. So I'll go through this question a bit more. So nondisjunction is essentially, it occurs at anaphase, so therefore it can't be red or green. And it occurs at anaphase because it's the stage where the spindle fibers start to shorten and pull your chromosomes apart. So for this question, you're mainly differentiating between yellow and blue, or anaphase 1 and anaphase 2. And the reason why it's anaphase 1 and not anaphase 2 is because if nondisjunction occurs in anaphase 1, that essentially occurs earlier on in the process. And if it occurs earlier on in the process, that means that you're going to get more widespread effects. So essentially your 4 gamete cells at the end, it's going to look like no chromosomes, and then double the chromosomes and double the chromosomes. Whereas if it occurred in anaphase 2, your first 2 gamete cells would have the correct number of chromosomes or the correct number of DNA material, and then your other 2, one would have double the amount and the other one would have no DNA material. So therefore, the worst outcomes will occur if nondisjunction happens in anaphase 1. Okay, yes. We don't actually know how much he benches. This was just a random guess.
200. Easy.
But,
easy.
Apparently one of my friends, I think a friend's friend goes to the same gym as Microfilaments. So maybe we can find out one day. Whoever picked 10 kilos, I show some
Disappointing.
respect. Show some respect.
Such disrespect.
Yeah, we'll go through it, Shreya. I will pull up the diagram and share screens after this question.
Okay, so wait, Peter, are you going through this or a different person?
I'll go through anaphase 1 and 2 first.
Okay.
So I'll share screen.
Yeah, sure.
Okay, so if everyone can see my screen right now, I have a diagram of what happens if nondisjunction happens in anaphase 1 versus anaphase 2. So on the left here, we have what will occur if it happens during anaphase 1, and then on the right, we can see what happens if nondisjunction occurs at anaphase 2. So considering the scenario on the left, if there's nondisjunction between these 2 homologous chromosomes, that means that one cell will get double the amount and the other cell will get none. And therefore, once this cell with nothing splits, the resulting gametes have nothing and nothing. So this is bad and bad. And then if we consider the other set of cells, this one already has double. So therefore, if it splits, it still has double. So this here and this here, bad and bad. So in the case of nondisjunction occurring in anaphase 1, you're going to get all 4 gametes being essentially defective. If it occurs in anaphase 2, however, one of your pathways is going to be okay. So you're going to have the correct amount of DNA in this cell, and therefore once it further splits into 2 gametes, you're going to have a good amount of DNA and a good amount of DNA. So that's good and good. However, the same situation happens with your other cell where it received too much. And, if this single cell here doesn't split properly, that means your resulting 2 gametes, one will have double and one will have not enough. So therefore, if you look at the end results for if non-disjunction occurs at anaphase 1 versus anaphase 2, you're going to see that anaphase 2 results in more favorable outcomes, as you have 50% being normal compared to if non-disjunction occurred in anaphase 1. So hopefully that helps you guys out. Explaining the other question, which question was it? Patient presents to ED with severe blood loss.
Oh, I'll explain that, yeah. Let me share screen. Okay, so when you have severe blood loss, you usually infuse saline solution because saline solution is the only solution which allows water to move into your cells. If you put in dextrose solution, water will actually move out over time. So usually you only give dextrose solution for people who are experiencing dehydration, because at that point in time, that patient is actually hypoglycemic and they have low blood sugar. So that's why they give dextrose. So that's the only time when you give dextrose.
Yeah, and just to add on to that, like saline is also much faster in raising like extracellular blood pressure compared to dextrose, which takes a bit of time to actually increase the BP.
Okay. Moving on. For now, you just need to remember that there are certain ions which have higher concentration inside the cell, and some ions have higher concentration outside of the cell. So Cl- is much higher outside the cell.
Yep. Outside the cell, you can kind of think as salt water. So outside the cell, you primarily have the sodium and chlorine ions, whereas inside your cell, you mostly have potassium and some negative protein anions. So this was the question I was like struggling writing because it has like it had negatives when I was writing it, and it just confused the heck out of me. But essentially it can't be, so apoptosis is programmed cell death. So if it's programmed cell death, that means, if it's programmed cell death, that means that the body has kind of planned for it, and therefore it's not going to cause harm to the rest of the body. So therefore, green is the correct answer because it is false. Yep. It's option blue because the cell kind of shrinks as the cytoplasm condenses, as well as the organelles. RJ, you're sharing screen, bro.
I know.
And it can't be yellow. Option yellow is incorrect because cell membrane integrity is maintained as your apoptotic bodies are formed.
Okay. Next one.
So this one is knowing the 3 different types of DNA repair. And excision repair essentially occurs to maintain the health of the cell during its everyday life. So what I mean by that is during the cell's everyday life, it's going to suffer nuclear damage, so radiation, or chemical damage. And if that DNA is damaged, you're going to need things that repair it. So that's what we call excision repair, and that occurs during the whole cell life. Other types of repair are proofreading and mismatch. So proofreading occurs during replication. So as your DNA polymerase is moving along, if mistakes are made, proofreading will occur. So therefore it can't be red. And it can't be yellow because that's what happens through mismatch repair. So that occurs right after replication, typically during the S phase checkpoint before it moves on to G2. And right before DNA replication isn't correct. Yeah. Ooh, Genesis. Yep, so it's not red. That's the main answer to differentiate from, because although it's true in some sense, it's not the best answer, unfortunately. So the resources that would have gone to the other bodies or the other gametes are kind of all condensed into one cell. Blue, meiosis generally takes the same amount of time, and green is incorrect. Yep, this is kind of a fact that you just simply need to know. This is what we were talking about before with the checkpoints and your cyclins and your CDKs. Yeah, that's just a fact. But don't worry, this one isn't too high yield. They might bring it up maybe once, but don't be too concerned about these questions. Nice.
All right.
Do you want to take this, Audrey? Or?
No, go for it. Go for it.
Okay. So it's not red because they have to be competent. So they have to be able to understand all the content. It can't be blue because blue, although it's a way valid consent can be given, it's not the 3 elements required for valid consent. So a bit of a confusing question there. And yeah, yellow is correct because number one, the person has to voluntarily agree, so they can't be coerced in any way. They have to be informed, so they have to know about, say, the risks, benefits, alternative treatments, what the procedure is about and what's going to happen. And they must have capacity because they have to understand and remember and be able to kind of explain it back to you. And that's all required for valid consent. Whoa, 26 correct answers in a row. Impressive.
Oof.
This one is simply a fact. It's a small detail in that one citric acid cycle will produce, it'll produce those quantities, 3 NADH, 1 FADH2, 2 CO2, but it won't produce ATP. It produces GTP, so guanine triphosphate, instead of adenine triphosphate. Nice. So,
Yeah.
sorry.
So,
yeah, you go.
So I'll just explain why the other options are wrong. So each amino acid can be coded by more than just one type of codon. There's like many combinations. That's the red one. Then for blue, it's not parallel, it's actually anti-parallel. And the third base is actually the weak. It's not that important. The first 2 bases are the most important. And yeah, so since you know that more than one type of triplet can code for a codon, that means that there can be several combinations for the stop codon.
Great. And last question, last real question. Nice. So this one, so as we know in DNA, adenine pairs with thymine and cytosine pairs with guanine. So therefore, if 30% was adenine, that means 30% must be thymine. So that gives a total of 60%. And the remaining 40% we divide in 2 to go between cytosine and guanine. So therefore, guanine would take up 20% of a person's nitrogenous bases. And then last final question. I'll be a bit sad if you guys don't get this. Don't worry, we won't cry. Nice. Okay, sweet. Does anyone have any questions from the whole Kahoot? So cholesterol maintains fluidity at different temperatures, essentially because so your phospholipid bilayer has fatty acid tails. And if we think about those fatty acid tails, they're essentially oil. You can think of it as oil. And what happens to oil at really high temperatures and really low temperatures is quite different. So at high temperatures, oil tends to become less viscous, so it becomes more water-like and it can flow around easier. And then at cold temperatures or at lower temperatures, like all oils do or fats do, they tend to freeze up. So our cholesterol serves to avoid this whole situation by kind of sticking together our fatty acid tails at high temperatures, so it prevents it from getting too liquidy. And then at low temperatures, it stops the fatty acid tails getting too close to each other, kind of prevents your dispersion forces and therefore stops your phospholipid bilayer freezing and becoming stiff and not working.
Okay, so now we'll do week 3 content. If anyone doesn't have any more questions.
Do you want to take like a 5-minute break for everyone to like chill out, or do you guys want to like smash through it? Soldier on. No, we do not need to know the function of the neoplasm. So for independent assortment, I can share screens for this one. So if we consider our cell, hang on, can you guys see this whiteboard? I can't see the chat.
Oh no, yeah, it's good.
Yeah,
It's good.
Yeah.
Okay, sweet. So if we use red as like one set of chromosomes and then green as another set of chromosomes, we essentially have random assortment at the metaphase plate when we have kind of like a mixture like this. So instead of it all being red on the left side and all green on the right side, we have random assortment in this fashion or something like this, so that we kind of get a mix of chromosomes in each daughter cell. Does that help explain what random assortment is? Fantastic. So after chylomicrons are packaged in cells, they're sent to lymphatic vessels. Yep, so how do they move into the bloodstream from there? I believe we're going to cover that today. Is that right, RJ?
Yeah, we'll cover that.
LDL transport. Yep, so
Yeah.
we'll go through that, Robin. No, what Elaine's saying, what you're saying with lymph draining into the venous system is correct. The lymph does drain into the heart after it goes around. So yes, that's a true fact. That does happen. Okay, do you want to get started on week 3, RJ?
Okay, sure.
Oh yeah, whoops. These slides are already up on the drive. Week 3 slides are up on the drive. So you can follow along either on RJ's shared screen, or you can bring them up on your own slide. Just make sure to use your Monash Uni email account, otherwise you won't have access. And we will not accept access from your private emails, as some of you would have found out if I emailed you. But yeah.
Okay, so I'll start with week 3 content. I'll start with lipid metabolism. Okay, so you just need to know the general structure of the different types of lipids. So there's TAGs, phospholipids, and steroids. So TAGs is just fatty acids, and so your TAG can be saturated or unsaturated. So what does that mean? It just means whether there's a double bond or no CC double bond. Okay, so if you have a saturated bond, it just means that there can be close packing of this TAG. So for phospholipids, you have a phosphate group and a variable R group. And instead of having, so for TAG, you have 3 fatty acids plus a glycerol. For the phospholipid, you only have 2 fatty acids plus a glycerol. And for steroids, you just have a fused hydrocarbon ring. And you just need to know that one example is cholesterol. So this is digestion of fats. Basically, when fats are digested, they're first broken down into the emulsified. So basically, they're made into small balls, like to increase the surface area using bile. And these form small micelles, which are like droplets. It's basically just to increase the surface area. And after that, enzymes work on them. You have lipases, which hydrolyze these lipids to form monoglycerides and fatty acids, basically breaking it down into glycerides and fatty acids. And then they are absorbed as chylomicrons in the small intestine, enter the lymph. And once they diffuse into epithelial cells, they are reconverted into TAGs. So someone was asking about how it moves from the lymph to the bloodstream. So basically, how it moves is when these chylomicrons enter the lymph node, there's actually the thoracic duct as well as your. There's a few ducts in your body where the lymph fluid actually enters your blood fluid. Okay, but you don't have to learn this for this chapter. You probably have to learn it for immunology. But it's just some good knowledge you can just know now itself. Okay, so for mobilization of fats, fats are stored in adipose tissue, mainly stored in adipose tissue, and lipases break them down. So this is mediated by glucagon and adrenaline. So you have fatty acids. Sorry, it's not fatty. They're not fatty acids unbroken down. It's TAGs are broken down into fatty acids and glycerol, and they're transported using albumin. It's just a transporter through the bloodstream, and it's converted, and your glycerol is converted to DHAP, which is used for glycolysis. So how does the metabolism of fatty acids actually work? So firstly, you need to know that it occurs in the mitochondrial matrix. And how it works is that there's a few stages. Your first stage is beta oxidation, where 2 carbons are cleaved off at each point. And these 2 carbons produce acetyl-CoA, which enters the citric acid cycle. So if you have whenever you have these carbons which are cleaved, there are 4 electrons which are released. And ultimately, you just need to know at the end of it, you produce 1 NADH plus 1 FADH2 per breakdown of the 2 carbons. Okay, so this is lipoproteins. Basically, your lipids are transported throughout your body using lipoproteins. So this table actually just summarizes the main things that you need to know for the different lipoproteins. So chylomicrons, you need to know that it's the highest in TAG content, and it transports after meals. And VLDLs, they transport TAG from liver to tissues, and it's between meals. LDLs has the highest cholesterol content and transports cholesterol ultimately to the liver, but around the body first. And this is the worst kind of lipoprotein, because this actually is involved in atherosclerosis if it stays in the bloodstream for too long. And you have your HDL, which collects cholesterol in the bloodstream and returns it to the liver. So this is actually like the good lipoprotein. So, and this is protein rich. So this is a good diagram where it helps you understand the whole flow. So chylomicrons transport lipids from the intestine to the capillaries, and the chylomicron remnants then travel back to the liver. Any free fatty acids which lie in your tissues and need to be transported are then transported using VLDLs, and the remnants will then be brought back to the liver. Some of them don't end up reaching back to the liver and are then converted to LDLs. And so these LDLs end up being stored in your bloodstream, and they attach to the walls, and this causes atherosclerosis. And your HDLs, they come from your liver, and they basically remove all these LDLs. It's sort of like a reverse cholesterol transport, and then bring it back to the liver. So that's why HDL is like the good lipoprotein. So lipoprotein uptake into cells. So every cells have these things called like Ldl receptors, and they're produced by the RER and Golgi body and are placed on the surface. So your Ldl receptors bind with APOP 100. You just need to know that it's a lipoprotein. And so once your LDL particle fuses, attaches to this receptor, it's then endocytosed and fused with a lysosome. And then enzymes then degrade it, and they produce amino acids, fatty acids, and cholesterol. So now fatty acid synthesis, it occurs in many tissues, mainly the liver and adipose tissue, but that's on the main side. And this only usually happens when there's excess energy. So instead of entering the citric acid cycle to generate ATP, acetyl-CoA is then converted to fatty acid instead. So you need to know there's three ways of regulating this. Firstly, hormonal. So insulin actually increases fatty acid synthesis because there's too much of energy in your body. And glucagon actually decreases fatty acid synthesis because you want to. Glucagon is usually released when you need more energy. Okay, so this whole process is actually end product inhibition. And finally, you need to know that if you have a high carbohydrate, low fat diet, this actually increases fatty acid synthesis. Low carb, high fat diet actually decreases fatty acid synthesis. So fatty acid synthesis is a complicated process. You don't need to know the entire process, but you need to know the first committed stage is the conversion of acetyl-CoA into malonyl-CoA. So this process requires ATP and is considered a committed step because this acetyl-CoA could be used for a variety of things, but once it's converted to malonyl-CoA, it's almost certainly only used for fatty acid synthesis. So the second step is of elongation, and this occurs using acetyl-CoA, which extends the chain 2 carbons at a time. So ATP, 1 ATP and 2 NADPH, again, you need to know this, are needed for each elongation. So if you want to synthesize a 16 carbon fatty acid, you'll need 8 CoA because you know the first step requires acetyl-CoA as well. And plus you have 7 ATP and 14 NADPH. So this diagram helps you better understand it. So you have your 7 acetyl-CoA, which is converted to malonyl-CoA, and you have an extra acetyl-CoA, so that makes it 8, and you have 14 NADPH, which is used as well. TAG synthesis occurs in the liver and adipocytes, but it only occurs during glycolysis. The reason why is because in the liver, glycerol is converted to glycerol 3-phosphate by glycerol kinase, and in adipocyte cells, DHAP, which is an intermediary of glycolysis, is converted into glycerol 3-phosphate. So this DHAP, which is an intermediate in glycolysis pathway, just shows the need for an active glycolytic pathway in order to synthesize TAG. So phospholipid synthesis. Again, you don't need to know the process. You just need to know that all animal cells, except red blood cells, can synthesize can synthesize phospholipids, and it's used primarily for membrane structure. two fatty acids are attached to glycerol 3-phosphate, producing a phosphodate, producing an acid, and then hair group is added to it, so it becomes phospholipid. Okay, so ketone synthesis. This is made in the liver from acetyl-CoA and occurs when there's low blood sugar. Because ketone synthesis is only used, you only require ketones when you're in starvation mode. And this process occurs when beta oxidation is occurring, and it produces 3 types of ketones. And you will study in next week when you require ketone and which organs actually use ketones. Cholesterol synthesis. Cholesterol is synthesized in the gonads, liver, and adrenal cortex. You must remember it's adrenal cortex and not adrenal medulla. And a similar amount is synthesized by the body daily, as well as absorbed from your diet daily. So again, the committed step is converting acetyl-CoA into HMG-CoA. And so again, cholesterol synthesis, you don't need to know the entire process. You just need to know the very first step. And that's shown here. So you need to know that when this enzyme, HMG-CoA reductase, is the committed step, and it's a rate limiting step. And it's controlled by glucagon. So glucagon restricts it, insulin increases the rate, and there's also end product inhibition. Okay, so cholesterol structure, you know it's 4 rings, and the function is that it's part of the cell membrane, and it's precursor for many things such as bowel, steroids, vitamins, and cell signaling, and it enters the cell via receptor-mediated endocytosis. So essential fatty acids, they basically cannot be produced by the body, so they must be absorbed through the diet. There are 2 types, mainly omega-3 and omega-6. So omega-3 is linolenic. It's the most common type in diet. And omega-6 are precursor for longer fatty acids, which are used for cell membranes and eicosanoids. And eicosanoids are basically paracrine hormones that are responsible for inflammation. And you'll learn more about this in immunology and later chapters as well. Okay, so now moving on to ethics. Who's doing ethics?
David. David.
Hello. Yes. Hello. I'm back. All right.
David, are you going to share your screen?
Yeah, I'll share my one.
Okay.
Before we get started on ethics, does anyone have questions about lipid metabolism? No, get started, David.
Okay, we'll answer that later. All right, so introduction to ethics. Ethics, like usually you would have had 2 ethics tutorials when they will be covering the 4 pillars of ethics and some ways to think about like the philosophical ideas behind ethics and why it's important. I think it's also just important for you guys to know that ethics is quite essential for your later medical practice, as it will allow you to make better clinical decisions and in more complicated times. For example, recently in the news, I'm sure everyone's been following with like coronavirus and COVID-19, that a lot of people are saying in Italy, oh, like why aren't they even giving ventilators to 65-year-olds like and above? Is it because they're just basically based on survival rates, and there's a lot of other ethical issues. So as much as this might feel dry and it's not very sciencey, it is quite important for you guys to have a good ethical understanding and be able to argue your clinical decision through these ethical principles. All right, so what are ethics? Essentially, ethics are just a way for us to understand things and solve these complex issues. A lot of times, as like what I said before in the example about COVID-19, there aren't like clear answers. There's no straightforward kind of things there. So we need to think about, all right, so what is a way to actually systematically deal with it, right? Like we always want to be systematically solving the problem. So there are 4 major pillars of ethics. I'm sure everyone knows this. So autonomy, beneficence, non-maleficence, and justice. So they all provide us a way to think about these problems in like a changing context. And things are always adapting. They're never static, they're dynamic. So your pillars are able to withstand that and still guide you through these tricky scenarios. So I guess your first assignment will be just to use your first assignment is to use these 4 pillars of ethics to help you solve like an ethically difficult situation. Have you guys had your ethics tune yet? Or like, is that coming up next week? No, okay. And coming up on Monday. Yeah, okay. So so this is a really good rundown, I guess, to what you guys should be looking out for in your ethics shoots as well. They'll be talking about the assignments too, I'm sure. All right, let's just jump into it. And yeah, this is recorded, Eugene. So autonomy. What is autonomy? Well, it's just respecting the rights of the patients. It's rooted in the traditions of freedom and having the liberty. So there's 2 sides of it. So firstly, it's like negative obligation. So we're not putting any restrictions upon other people, and positive obligation, right? So it provides treatment that fosters autonomous decision. So we want to promote that people, we want to promote this idea that it is your decision. So for patients to have the right to autonomy, they must be fully informed and not under any external influences in making the voluntary decision. Of course, as the GP or the doctor that you will give some guidance. But at the end of the day, your job as a doctor is to give the right amount of information and lead them to the best decision-making capacity for themselves. And everyone is saying like, okay, autonomy is good for the patient, so they'll think for themselves. But another important part of autonomy is that it also protects you. So later, when the patient is talking about, oh, like why didn't you tell me this or that, or like why didn't you inform me about this? You as a doctor can be saying that no, you have full autonomy, and I did explain anything. So autonomy is actually a pillar of ethics that does protect the medical practitioner as well. Yeah, so we're not 100% sure about your assignment. I'm sure just like listen to what Richard will say, or like whatever faculty says. Great. So beneficence. What does beneficence mean? So it means that as a doctor, you should be doing what is the best thing for the patient. What is good for the patient? You should be doing the outcome. So ultimately, you need to balance the risks of procedures to the benefits. And this principle essentially just encourages you as the doctors or the medical practitioners to have the patients get the most out of it. So let's just say like you have acne, right? And you want to get rid of the acne. So the doctor's like, okay, you can have doxycycline. It's a drug that you guys will come across later on. So this drug, but the side effects is photosensitivity. So if you're like a really avid tennis player, then because I don't want to do any, I want to make sure that your acne gets good, but I also have to balance it to the risks of you getting sunburned easily under the sun as a tennis player, then I might try and like get another drug. So these ethical principles really guide the way that you think and finding the best tailored approach in helping that specific individual patient. Now, non-maleficence versus beneficence. So we're really talking about beneficence, how it's like doing the best good for the patient. But non-maleficence is like you not doing any harm. Evidently, we will need to balance the two in a more topical issue with like COVID-19. A lot of the like drugs that's using are very taxing on the body. There's a lot of morphine, a lot of opioids usage, but that's decreasing the pain, but it's also not good for them in the long run. So it's just trying to balance these two, which is one of the big parts of ethics. And finally, justice. What is justice? Well, so justice is essentially doing the things in favor of the wider population. Appropriate distribution of resources to those who need it, and in regards for social characteristics. This is one of the, I guess, different parts of almost the different parts of ethics, because it's more so looking at it at a society kind of perspective, rather than looking at it very specifically into like that individual patient. So we're saying that this goes back to the patients in Italy, how trying to judge which patients to save and which not. And ultimately that's a difficult decision, but justice does play a major component into it. So when we always want to be talking about informed consent. So what is an informed consent and what components do you need? So to have an informed consent, you need to be able to make an informed decision in which you need to have the capacity. Are you of appropriate mental capacity? So that means you're not under the influence of like drugs. Yeah, same. Like the slides are glitchy for me on the on my other monitor. I might.
Wait, no, it's better now. Go back one. Or do you want me to share, David?
Let me just. How do I exit? Yeah. Yeah. Do you want to share actually?
Yeah, I'll share. All right, there you go.
Thanks. Yeah. So components of informed consent, you can't be under drugs or alcohol, or if you have very severe mental health issues that will impair your decision making. So it's pretty self-explanatory in why you would need to have that capacity. Redo justice, yeah. Okay, Peter, do you want to go back one slide?
Yeah.
So justice, okay, what I said about justice was basically, this is one of the different ethical principles in that it's not individually talking about specific patients. Beneficence, non-maleficence, and autonomy were always like thinking about the well-being of that patient. But justice is more so on a larger scale. We're thinking about the society as a whole. So for example, this drug, it's very, very limited, and we have to be thinking, like, do I want to be giving it to everyone? No, like I want to be giving it to the patient who are most sick. Or for example, ethically, if a patient has coronavirus or has COVID in the community, then it is a part of the justice principle in which we need to say, all right, we need to isolate them, otherwise they're going to be doing a lot of damage to other people. So justice is think about it in a wider population and think about why everyone needs to have the same amount of access to resources rather than those coming from a more privileged background. HKS and public health, a big part of it is based on this principle of justice. So definitely listen for this actually in your HKS tutorials. All right. Yep. So components of informed consent, I've talked about capacity. Informed consent, there's just a few things. So procedure, you have to talk about the procedure, the risks, the alternatives, the outcomes, and the possible complications. You guys will actually go into what the components of a consent later on in your study, so I won't go too much here. It's not required of you to know the exact components of that consent. You just need to know what formulates informed consent. So understanding the information given. So obviously if you say like, hello, we're going to extubate your mother using a hypographine-based solution of 600 millismolarity. Like, firstly, that makes no medical sense. Like, it doesn't mean anything. But obviously, like, you can tell that this is difficult to understand, and it's also gibberish. So you're going to make sure your patient understands. So you can be speaking a lower register by telling, it's like, okay, so we will be removing the tube from your mom because she's getting better, right? Like, so you want to communicate it in a way that's more direct. And voluntary, don't ever like almost hold someone at a gunpoint, always saying like, oh, if you don't do this, I'm not going to treat you anymore. So you should be doing it and fostering the idea that it should be a voluntary consent, which takes us back to the autonomy principle. Yep, just next slide. Medical paternalism. This is what we were understanding last week or the week before when we were talking about epistemological ideas of how we can understand medicine. So medical paternalism back in the day is just like, I'm the doctor, so you got to listen to what I say. That's what it used to be like. All the patients used to just listen to whatever the doctor said, even if it was almost a selfish decision driven by external influences, for example, pharmaceutical bribings or things like that. So when we think about this, we have to think clearly about if the doctor is giving the patient the full choice. So it's overriding a patient's preference and autonomy to when they believe it is in their best interest. So there's two types of paternalism. The first one's weak paternalism, which is good. So you don't have the mental capacity, or at the same time, you're too tired and you actually do trust in the doctor to make the best decision for you. The doctor can help you and guide you. However, it's got to be aligned with the patient's best interests. Like, you've got to be standing or trying to stand in the patient's shoes and consider what is important to them and making the best decision. A lot of like Asian communities actually really prefer weak paternalism, as they feel like that's the role of like the doctor in a traditional setting. However, more and more so, that's also becoming westernized into have more autonomy. So in strong paternalism, often it's like just when the doctor overrides the patient's voluntary informed medical decision regarding the treatment. That's just forced. There's no flexibility or freedom there. And confidentiality, this is, I'm sure all of you have practiced this in your, I think you guys call it a OSE, is that right? Like your new CSSE or whatever it's called. So OCES, okay, yeah. So all of these, OSCEs, okay. OSCEs, all right, yeah. So confidentiality, I wanted to show you that everything we talk about today will only be communicated between you, myself, as well as the treating team, unless otherwise stated by law. So it always comes up really smoothly off the tongue, and you'll know this really well. And it's really important just to give people a certainty and assurance that all this information will be secure and won't be leaked out. A lot of these situations, I'm sure, in like your NOI situations, you guys might have come across this as well. For example, a young patient. That's not even the fastest we can go, but we can do a challenge for this like later on in the year. Yes, so for example, a teenage girl comes in saying that she is pregnant. So it is really important for us to think about confidentiality and not breach that trust, because otherwise it will actually endanger and risk the welfare and health outcomes for that patient. So confidentiality, therefore, is implicit in all therapeutic relationships and is essential in building rapport. So it's just making a good connection. And like, if you can't keep a secret, like, man, you just gotta up your skill, and your character. It's a good thing to do in general. And some exceptions, though, for breaking confidentiality is, firstly, exceptions are allowed from the initial party. So if the pregnant young lady comes in and tells you, like, okay, you can tell this person, then you're allowed to tell them on their behalf. Obligation to prevent harm to another individual. So if someone comes in to tell you that they have like coronavirus and blah, 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 then obviously things like this, they will be harming other people, and it will be usually requested by law that you have to report it. There is a list of reportable diseases, including HIV, amongst other things, that must be reported within a certain amount of time frame. You guys can look into the details of that. It changes year to year as to the exact disease that they will choose for your exam, as they did with us. Next slide, Peter. Thanks. So types of ethics, I'll just go briefly into this. So consequentialism basically means that you judge what happens based on the outcome. So let's say you're investing stocks in the market and you put in $500, but you lose it all. So you say that's a bad decision, but you don't know, like, so you're judging using the outcome to justify your decision. Whereas rights-based theory, you can be like, okay, so it's assumed that free speech, civil rights, access to food, and right to life and healthcare are quintessential aspects that we must uphold. So you can't ever withdraw from that. So it's just based on these values that we all share, and it's often like really like the United Nations Charter of Human Rights and all of these things. So that's called deontological. Virtual ethics argues that a person who is virtuous will make the most reasonable decision in certain situations. So this is based on characters, including like mercy, honesty, fairness, integrity, fidelity, generosity. So all of these things are characteristics. However, one of the downfalls of virtual ethics is that it is harder to like quantify as in what is mercy, what is honesty. Whereas rights-based ethics allows you to say freedom of speech means you can say a lot of things. Yes, consequentialism is the same as utilitarianism. Utilitarianism is just an old word we used to use in Kantian philosophy. So that's a guy called Kant. Just you can look him up on YouTube. There's like a bajillion videos from like School of Life and things like that. Yeah. Next slide. Done. All right. Thanks, guys.
Okay, so I'm going to be doing medical history, and I'll take you through HKS as well. Meta history, I'm just going to do a really brief overview because obviously the video broke, but like we're going to be filming another one tomorrow, and hopefully it doesn't break this time, and Jating and I will hopefully be able to upload that very soon. Okay, let's go. Where's the chat? Okay, I'll keep the chat open. Just like a question, are you guys like having CSSEs or OCEs or whatever they're called? Like, are you guys actually having them this semester? Oh, okay, so they're still happening. Okay. Oh, news. Okay. It's going to be interesting for everyone. Okay, so I'll just quickly go through it. So basically, this is an overview of the full interview. Okay, so obviously you start with identifying data, making sure you've like got the right patient, because it's a little bit awkward if like you start the interview and then like you like realize halfway through that you don't actually have the right patient that you were supposed to go with. And then history of presenting complaint, that's kind of the key bit. That's kind of the key bit that you guys want to learn at this point. Hi, Jane. Hi. Yeah, so that's kind of the key bit that we want to you want to be able to focus on at the moment. Systems review, you can look forward to that next semester. Basically, that's so like what directly leads on from a history of presenting complaint is actually like doing like full examinations and like actually like going in and like doing stuff. But don't worry, it's not semester one. And then past medical history, family, drugs, and social history. A lot of this seems like a lot of rote learning at the start, but you'll find that like the more practice you do, and the more kind of role plays you do with other people, it'll become like second nature. So especially with HOPC, like a lot of you learn like so many acronyms about how to like memorize key questions that you need to ask. But in the future, it's going to become like, you'll just naturally follow up on symptoms. Like they'll say like, oh, you know, like I've been getting a lot of nausea, and like you'll automatically go, okay, like, you know, how long have you had it? Are you like, do you have dizziness as well? Like, you know, how did it start? Do you have dizziness? Do you have like ringing in your ears? Do you have like, so it all comes like, it seems really hard at the start. But yeah, it all like comes together in the end. Sorry, I'm just like looking at the chat at the same time. Oh yeah, and you'll have to start getting used to like using abbreviations a lot, because you know, taking notes while you're taking a medical history is also another skill that you're going to have to like start learning, and it's like, yeah, have to like keep up. Oh my god, what did I? Oh wait, okay. Do people want me to go through like WWQ, like HOPC again? Because I went through it last time. Yes, no, maybe.
We can just include it in the video tomorrow.
Yeah, okay. So basically, past medical history, you need to go through any current medical conditions they have. A lot of people do forget to ask that because they think it's all focused on like what they've had in the past before, but like you need to know what they also like currently have been diagnosed with. Surgical history, hospitalizations, they kind of like go hand in hand. Any injuries, both past and like whether they've had them recently. Childhood illnesses, a lot of the time they don't remember whether they've had childhood illnesses or not, but just ask it just to be sure. Immunizations, just ask, are your immunizations up to date? And make sure that you ask whether they've actually experienced their presenting complaint before. So if they come to you and they go, oh, you know, I have like really bad back pain, and then you can just ask, okay, so like, have you had this kind of back pain before? Or yeah, so just make sure that you double check that they haven't experienced it before. Family history, this one's kind of self-explanatory. You basically just ask about first-degree relatives, so that's parents, siblings, children, and just ask whether they know if there's any like kind of genetic conditions that actually run in the family. Sometimes they won't know this as well, but just asked to be sure. And if their parents have passed away, just follow up on cause of death and age as well, especially if the age that they pass away at is a little bit like suspicious. Like if they pass away in their 60s, you know, it wouldn't have been due to like old age, it would have been to another reason. Medication and allergy history. This is kind of we have acronyms to remember basically what you need to follow up on. So we usually use the drugs mnemonic here. So any doctor prescribed medication, recreational drugs, so alcohol, smoking, and actual like drugs. User, so that's like over-the-counter medication, alternative medications. That's kind of like, so over-the-counter like from the pharmacy, alternative like your CAMs, so like acupuncture, massage therapy, aromatherapy, or whatever. Gynecological, OCPs, hormone replacement. This is like you need to remember to ask about, okay, OCP stand for oral contraceptive pills, by the way. Do remember to ask that, especially for female patients, because in real life, like female patients don't associate OCPs with drugs. Like when you ask them, oh, like, are you taking any medication? They won't think of them taking the pill as them being on medications. So like for some reason, they just like forget about it. So just make sure that you ask about OCPs and hormone replacement if they're like postmenopausal as well. And just sensitivity. So do they have any side effects when they're taking the drugs? So drug, alcohol, smoking history. The key thing just to remember is just, oh, what the hell? Okay, give me one. What? Oh my God. Sorry, my laptop's just decided to. What? Okay, we're back. Just the key thing to remember is just be like non-judgmental when you're asking. You don't want to come across as like you're really like kind of judging them for like if they're like a heavy smoker, if they're a heavy drinker. With drugs, this is kind of what people usually like a key. This is what people just usually ask is just say, this is a question we have to ask everyone. Have you ever taken recreational drugs before? And then if they say yes, just say, when did you take them and how long were you taking them for? Alcohol, just do you drink and how many drinks? If they say yes, just ask them how many drinks usually and how many drinks when they're socializing. And if you feel that their drinking like is on like kind of the heavy side, remember to ask the cage questions. So basically it's, have you ever felt the need to cut down on your drinking? Do you ever feel annoyed when people ask you to cut down on your drinking? Do you ever feel guilty about your drinking? And have you ever needed an eye opener in the morning? An eye opener is basically just like the minute they wake up, they need like a drink in the morning to just like kind of start their day. And then smoking, you do need to ask, do you smoke? And also, have you ever smoked? Because they might not be a current smoker, but they might have been like a past smoker in the past. So you do need to follow up on that. Make sure you ask like what they smoke. Usually it's just cigarettes these days, but it could have been something different, especially if they're like older patients. And just like asking like how many cigarettes per day, you know, have you ever thought about quitting and have they actually quit? And then we move on to social history. Basically, this is based on the Craig-Hassard like essence model. Have you guys had your Craig-Hassard like essence? Not yet? Okay, well, you'll have it. Craig Hassett, he's Craig Hassett. But basically this is the model that covers all aspects of social history. So it's actually a really good way of just asking like holistically about like a patient's kind of life. So that's just like environment. So like, sorry, I'll come back to the questions by the way. So environment, basically that's just like work, home, living conditions. Make sure you ask who do they live with. Stresses, that's also like at work, at home, are they financially stable? Are there any like stresses with family, etc.? Spirituality, usually people just ask this kind of set question is, are you particularly religious or spiritual? I personally like don't understand why we ask it, but it's just a required question, so just ask it. Exercise, so do they exercise? How do they exercise? How frequent? You know, that kind of thing. Nutrition, that's basically just diet. Usually you can just ask like, oh, what does a typical meal look like for you? Or like, what does a typical day of like meals look like for you? Connectedness, that basically just means like support networks. So do you have like a good support network? Do you have people that you can rely on for support? So usually that's like friends, neighbors, family relationships. And then education, basically, that's actually focused on like their own medical health literacy. So just ask like, if you're sick, do you know who you can go to seek help? Like, do you know where to find your GP? Do you know where like the nearest hospital is? Do you know what to do? Okay, let me just look through the questions because we're going to move on to HKS. Are there any questions? Oh, let me just. Yeah, don't ask guys if they take OCPs because that's like, no.
And if you accidentally do, like, don't stress.
Yeah, it's okay. Like,
just be like, sorry, I meant to ask you, do you take steroids?
Oh, and also, if you do ask if, like, especially for drugs, make sure you ask them like what they're taking the drugs for. Just to be sure, like with doctor prescribed medications, they usually tell you like, oh, I take this drug for like my high blood pressure. I take this because I have diabetes or something. But like with OCPs, ask if they're sexually active as well. Because even though like some people like are taking it for acne, they take it because of like, you know, irregular periods or whatever, but just double check, like, are they sexually active? Okay. I'll go through health determinants next time then, because I feel like people are like, it's late.
Yeah. Does anyone else have any other questions about the medical history? Because it will turn out to be a very, very important part, if not the most important part. Faculty always goes on about how for most of your diagnoses, you're going to diagnose from their history. So being able to ask a thorough and comprehensive yet concise medical history is a really important skill. Yeah, but otherwise there's still more content for week 3. However, it is getting fairly late, and I feel like a lot of us are tired. So we'll probably stop it there. But if you do want to go ahead by yourself, before our next session, the slides are up on our Google Drive.
OCE, I think someone mentioned, yeah, observed clinical encounter. I don't know why they're changing it, but
yeah, med faculty likes to change names all the time.
yeah.
But the Shine-Dalgarno sequence is, sorry, this is a completely unrelated question, but we'll go through it anyway. So that's essentially just this, it's kind of like a promoter region, but for prokaryotes. So it's essentially just where the whole process starts off in prokaryotes.
How important is biochem? It's important.
Unfortunately.
Yeah.
I think it makes up majority of exam 1. Yeah.
It's like a huge chunk of exam 1 and then also a huge chunk of exam 2.
A mood. Yeah.
But just like you don't do much of it after that. Like basically it's like non-existent.
Yeah. Oh yeah, Sem 2 it's like biochem is just off
Yeah,
the
you
list.
just like don't do biochem ever again.
Yeah. Biochem, I think you can typically get by knowing the big idea of things.
Yeah.
Once you go through like practice exams as well, like you'll figure out keywords med faculty likes to use. So they're like buzzwords where as soon as you kind of see that question or see that word in the question, you know exactly what the answer is going to be.
Yeah. But the so for like passing first year, like I didn't know that much biochem. Like basically, like I walked into the exam, like the metabolism like thing. It was just like I knew like 3 enzymes, and that was like basically it. And I still passed, and I did fine. Like as long as you know like big ideas, you know like kind of what's going on, you'll be fine. Like it's okay. Also, like year 1 is like pass fail, so like year 1 and 2 is pass fail. So
honestly, like don't matter, like don't stress too much, especially since I didn't do bio, and so when I came to do like med straight away, I was so stressed. I was like, what are all of these things? Okazaki fragments, like what is this, right? But later on you just find like once you know everything in like the big picture of things, like everyone will end up drawing like this huge map for themselves for like biochem pathways. But just like make it yourself and understand like where things fit, and it's like usually understand the flow of things. And like if you're exercising, what happens? If you're fasting for 8 hours, what happens? Know those things more so than learning like, okay, after malonyl oxalate, it goes into aceto. Like I don't know, like I don't even remember it anymore. But if you ask me, like if you fast for 8 hours, like which state of digestion or like metabolism do you go into? That becomes better. Yeah.
Yeah, and after semester 1, anyone who did bio 3-4, you pretty much just lost your advantage.
yeah.
Yeah.
But yeah, anyone have any other questions? Doesn't have to be about content, just about like life in general.
Yeah,
like we're all like going like stir crazy because we don't like have any human contact. How do you survive med school?
You don't.
You don't. Just
Don't
just
catch
like
COVID.
Don't catch COVID.
Oh, Eugene, love you.
Oh, great friends.
Is sleep important?
Yes.
It
Oh my
is,
goodness,
but
it's so
also
important.
like if you like, yeah.
Honey, it is.
Yeah, there was
It
like
is,
one
though.
time.
It
There was
is.
like one time in, it was like Sem 2, and it was after mid-sem break, and like the first week back, I did not get enough sleep, and that week was probably the roughest week of like the whole of year 1.
Yeah.
And I say that's because I didn't get enough sleep. So definitely sleep.
Yeah.
Try to form good sleep habits. But that being said, I told myself I was going to sleep at 11 for this year.
That's
and last
so
night,
bullshit.
And last night I went to bed at 3. So
Is a good sleep schedule realistic? Yes, if you stick to it.
Yeah.
If you make yourself a promise and you stick to it, you can.
Yeah.
But all of us are like bad examples, like except for Zoe. Zoe's like a good example. She goes to bed at like 9 every
She's probably
day.
sleeping right now.
Yeah.
On the question of how do you memorize stuff, so like we use Anki, use spaced repetition, and active recall. So those are the 2 things that you should definitely be using in understanding like memorizing content. So do things periodically and consistently rather than like just cramming everything. Like you might be able to remember it the next day, but ultimately your long-term retention of your memory will be much worse. So use Anki, use Quizlet, use whatever flashcard thing you'd like to use. There's a lot of videos online. We'll link them on our Facebook page. Not sponsored, so make sure you watch it. So yeah, I think it's just like routinely and strategically going through it.
Yeah. In addition to that, I think even just like asking questions to your tutors or asking questions to your friends, I think, or even just like people you don't know in the cohort, since you guys haven't really met for real, I think that's really, really useful. Like we still use that this year during our active learning sessions whenever we have a worksheet to go through. Normally like a bunch of us call together or like form some collective answering document, and like that helps us, like it helps the stupid amount. And that's like if your study group is productive.
After the exam. Yeah, after the exam, like you'll find that all the content just goes like, it's just like, it's like gone.
Yeah.
Also, someone mentioned like clubs and committees. I think we have like a really wide spread in this PSP. Like, like we can list them for you. Like we can go through them.
Heini, what are you in?
So I'm on Vespa. So everyone has to come to Vespa now because you're all obliged to. So please come to Vespa. We're trying to do it online. I don't know if it's going to work, but like we'll try. But Vespa's really great. You basically like work through cases from like year 1 to 5. Oh yeah, okay, I'll answer that. What's Vespa? So basically it's like we have like a case writing team, and basically it's really highly detailed clinical cases that are structured for years 1 to 5, and basically the year 4 and 5s will facilitate the case. So basically they'll just be running everything, and we just work together to solve. Like it'll be just someone who like, it's like the patient presents with this, and like you'll get like a couple of vital signs, and basically you have to work through the case together as a team. It's just like really great like revision. You get like a lot of clinical what's the word again?
Experience.
Yes, that. And basically, like, sorry, my brain's like a little bit dead, but like if it was in real life, you actually do get to like practice clinical examinations as well, and you get like a kind of sneak peek look into like future year level content as well. But yeah, it was supposed to be in real life and that didn't happen, but it's like it's moved online now.
And it's pretty funny, like, for example, the first one, like, they were talking about ABGs, so, like, obviously all the medics were like, oh yeah, arterial blood gas,
and all of us were just like,
all of us Japanese
ABG!
Like, Asian baby girl.
But yeah, so like you might feel like a little bit out of place at the start, but like you do learn a lot of content. We have like great summary notes that we give out at the end. Yeah, but it's really fun. Also, what did I say? I'm on Vespa. Also, Operation Smile. I don't know what's happening with Operation Smile, unfortunately. We were supposed to have the cocktail night, and that obviously can't go ahead at the moment, and that's like our main source of like fundraising income. So like I don't know what we're going to do anymore. But yeah, I know maybe we could do an online cocktail night. Everyone gets dressed up, but it's like not the same vibe, so whatever. But yeah, please come to Operation Smile events in the future.
Please come to music. Our applications are still open, so make sure you apply.
They close on Monday.
Music is the largest subcommittee in MUIS, where we cater for the entire student body, regardless where you are, even in Gippsland. And we have a bunch of activities, even with like UD kids, and we're heavily involved. So definitely get involved, and this year is like a terrific year because you have a bunch of time and you'll meet a bunch of committee members and get exposure to the surgical world.
Yeah, really good experience. What else is there?
Oh,
there are
MMO.
also like AMSA events. Oh yeah, MMO. AMSA runs a lot of events throughout the year. And I think the really good thing about AMSA I found was kind of linking up or getting to know other people from different unis and different year levels. And I think that's like really one of the only ways I got to know like some Melbourne uni kids, like some final years. And like, even if events do get cancelled, such as convention, I think the whole experience and getting to know different people is so worthwhile. So I would also encourage that as well. What other things do we have on? Anything?
Yeah, guys.
Muppets, TBH, guys.
This is a gene. The gene's our friend.
No, but like Teddy Bear Hospital is actually so fun.
Yeah.
Like
TBH is
it's
so fun.
amazing. Like I love Teddy Bear Hospital, but I don't like what's happening with Teddy Bear Hospital now.
We're just, keen. The next SEM is what we're doing. And like, think about how you like explain to kids what a liver is, or like blood pressure without using the word pressure. So that could be a homework.
Definitely try TBH. Like it's probably one of the most unique experiences as you'll have as a med student. And it's out there in the community, like actually interacting with people. So it's pretty good. And you also find out whether you like kids or not. So
Kids are so cute. The kids are so cute. Yeah, you do figure out if you want to do peds or not in the future. Like,
Come
join MMO, guys. If you're playing instrument, please come join MMO. MMO is like honestly best like vibes ever. It's so chill and it's so much fun. It's so sad that we can't like our sign-ups because it's not happening in semester 1 anymore. So it's like really sad. But I think sign-ups are going to open up again for semester 2. But we'll have to like double check with the committee and stuff.
Touch wood.
But no, MMO has better vibes. MMO's the best vibes. But yeah, we have like free food every single week. We just like chill and play like cool music. And we're like low-key like professional orchestra. So like, yes, we are. Rob is amazing. Rob is like adorable. He's just
there's also the committee if you like drama or theatre.
Med Review?
Med Review.
Yes, Med Review. They put on like a yearly show, which is great. You guys should all go watch it. I'm not in it because I can't act.
Yeah,
and I can't
none of
sing.
us.
So yeah. And also be sure to check out any external conferences that are held, such as ASSC, which stands for the Australasian Students Surgical Conference. Unfortunately, it's cancelled this year because COVID. Don't be shy. Don't be shy to put in your applications. Even though you're like just a first year, I put in my application for it at the end of first year last year. I'm not expecting to get anything, but I actually got onto the committee with a bunch of 3rd, 4th, 5th years from all across Australasia. So yeah, it's a great way to make connections with older med students as well.
Yeah. Jumping back to the questions, we're getting like a backlog thing. When is it okay to start forgetting stuff?
After exams, honestly,
yeah, from
guys.
personal
After
experience,
exams.
like after the Sem 2 exams, and we just went to holidays, I had like a streak thing on Anki. So like for the first few days, I was like, I have to do this. Like I have to just revise like 30 cards a day and it'll be fine. I'll keep the streak. But then I gave up and then I lost my streak and then I lost my motivation. So I would say definitely take a break over the holidays because hopefully if you've learned it well, it'll come back pretty quickly when you do start back at uni. Any other questions we've missed? AMSA, F in the chat. Thanks, guys.
Oh no, poor AMSA.
Our sign-ups for MMA. Music camp.
What else?
David, please run GHC. Yeah, bro. GHC
Bro, guys, that's
like
it's in our 4th year, so.
Yeah. Hopefully, I don't know. Wait, can you guys run? I don't know if you guys got the post from, like Jordan and Sarah, but they were calling out for members for the core team for Melbourne bids for GHC, which is Global Health Conference in 2022. I don't know if that's open to you guys, but hopefully it is. And if it is, go for it, I think. Although I don't know if applications are closed because
It should be
waiting for
subcom, but I'm sure if you just message Jordan, like he'll be super keen to.
Yeah. Otherwise, like subcoms will be opening up close to the date, probably next year sometime. But yeah, I think like you don't lose anything from applying. Like a lot of the time, you might think that you're not qualified to get in. but turns out you probably are. And you don't always have to know everything going into position, because a lot of it is like a learning process. So yeah, like submit the application. No one really has to know. You don't even have to tell anyone you're submitting it. And then later on, you just rock up promoting the post. And then like, oh, fancy you here. But yeah, it's always a good idea to apply for things. Oh. All good. Thanks guys for turning up on such short notice and on a Saturday night. Oh yeah, Scriblio. I forgot about that. Yeah. I don't know what the limit for Scriblio games are. Yeah, I actually have no clue what the limit for Scriblio games are. Hopefully we can fit like a decent chunk of you guys in. Oh, 12 people. Oh, sad. Oh, rip. Bro, I made like a list for like Scriblio words, like med related, that we could do. Screen
So we
share
would just
drawing.
run like 2 rooms or something.
2 rooms? Send the list. Actually, yeah, that's a good idea. I'll send the list in.
Wait, send it to us and then we can, I can host it.
Okay.
Like I can host, like we can host, how many of us are there? There's 30. So we can do like 3 rooms.
3 rooms? All right. Let me find the list. I haven't tested this list and whether we can actually draw these or not.
Nah, surely improvise.
Oh, some of this is also week 3 content, but whatever.
Oh wait, we can go into.
You guys can guess what you
pick
think it is.
You don't know what it is.
We can go into breakout rooms for this as well.
Oh, breakout rooms.
My laptop's gonna die, so I'm gonna head off.
All right, Tiny.
Bye.
Bye. I'm gonna stop recording now, I think. Yeah?
I think we can stop the recording.
Yeah. Hopefully it's recorded this all, because like, I know it says recording right now, but it better work.